First is lipid structure. So what are the different types of lipids? Main categories of lipids that you will have to know are called free fatty acids and glycerides. There's also a longer version of the name glycerides. It's with aldoglycerides, but either way, it means the same thing. There are also lipoproteins and glycoproteins. Those aren't as important, not in the sense of your body, but in the sense of the MCAT. They don't really ask questions about that. They may ask some general basic questions, and so if you want to look into that, you should, but it's not as directly important, it's not as crucial as some of the other stuff. So the structure of a free fatty acid is as follows. It has this long hydrocarbon chain of just carbons, hydrogens, nothing else, as well as this hydrophilic head. Can be a carboxylic acid with an OH here. In this case, it's not, it's bound to a sodium ion, right? But the main point is that it's a hydrophilic head with a long hydrocarbon chain as a tail. And there's a difference between, again, a free fatty acid and a glyceride. In this case, the monoglyceride, which it looks almost the same with the same basic structure, and then the head, but it has this glycerol as well attached, and that's what makes it a glyceride. That's where this part comes from. So in terms of saturated versus unsaturated, this is a common thing that you'll see with fatty acids. What you saw previously, this is called saturated, whereas this is called unsaturated. And the difference is that in this long chain, an unsaturated will have a double bond or multiple double bonds throughout its structure, whereas something that's saturated will not have any double bonds at all. It'll only have single bonds. The reason it's called saturated versus unsaturated is because if you imagine each carbon is not just carbon, it has hydrogens attached to it here and here and here, everywhere. A saturated one that doesn't have any double bonds is saturated fully with hydrogens, meaning that each carbon has exactly 2 hydrogens on it, except for this tail end, which has 3. And so it has the maximum number of hydrogens per carbons that it can possibly have. An unsaturated one has this double bond here, so all of these will have 2 like normal, but this carbon will only have 1 hydrogen each, because one of the bonds for each carbon is being used up to make the pi bond. This is results in it being unsaturated, and so it has less than the maximum number of hydrogens that it can have per carbon. That's just an easier way to help you remember it. Another important fact that you should probably know is that in general, unsaturated free fatty acids have a lower melting point than their saturated counterparts. Again, a big aspect of melting point is the size. Of course, if you have a longer chain, it'll have a different melting point than something that has a shorter chain. But in general, if you have the same length of the chain and a saturated versus an unsaturated, the unsaturated will have a lower melting point, meaning it will become liquid much easier than a saturated. Next, we want to talk about glycerides. So there's this molecule called glycerol that looks like this. And then you have these 3 separate fatty acids, and I'll actually do it like this to show you that they are 3 separate molecules, and each of them has this R group, and it's a different R group for each of them. It can be saturated, unsaturated, doesn't matter, and it can be different lengths. They just have this long chain, each coming off of them. And then these 3 plus this 1 glycerol actually can react together by virtue of these to form a triglyceride, which is a triester of glycerol. And you can see here, this is where the glycerol is. And this OH is actually a combination of this OH and this OH. So this is a dehydration reaction because you lose one OH and you lose an H to make an ester. And then you have just one O connecting these two carbons, right, to form an ester. And then all three of these chains are like that. So it has this really hydrophilic head, as long as these three really long hydrophobic tails now. An important reaction to know with triglycerides, and this doesn't really happen in the body, but I'm sure many of you learned this in general chemistry. It's called saponification, or the making of soap. If you know that originally soap was made just from animal fat, pig fat, usually, I think. And the reason it was is because you have this triglyceride from an animal, some fat from an animal, and you combine it with sodium hydroxide. And then in the end, you can, when you react these two, you end up getting the glycerol as well as the soap. Okay, and the three means that you get three different molecules of the soap. And if you notice, it looks very similar to a free fatty acid. It's just instead of an OH, it's now bound to a sodium. And this is how many very like old school traditional soaps were made in the old days. Now it's much more different, but it's an important reaction that you should know. Next, let's talk about beta oxidation. Beta oxidation is the main form of metabolism of free fatty acids in our body. It's the equivalent of glycolysis for carbohydrates. It creates a lot of NADH and FADH2, just like glycolysis in the Krebs cycle, but it also creates a lot of acetyl-CoA, which, if you remember, is actually the ingredient that can be used for the Krebs cycle. So when you have a carbohydrate, the way you make acetyl-CoA is you go through glycolysis, and then from glycolysis, you have pyruvate, and the pyruvate gets converted through pyruvate dehydrogenase into acetyl-CoA. With fats and lipids, the free fatty acid actually gets converted into many acetyl-CoA. Like I mentioned, free fatty acid could be very long chain, hydrocarbon chain, could be 16 carbons in length, and acetyl-CoA is only 2 carbons in length. So out of one fatty acid, you could actually make 8 acetyl-CoA, and each of those acetyl-CoA can go through the Krebs cycle, whereas if you have glucose, just carbohydrate, you can only make 2 acetyl-CoA and 2 cycles of the Krebs cycle. So this can be a lot more effective. Firstly, most of the food that you eat, like the fat itself, is in triglycerides. So that must be broken down into free fatty acids by the body, as only free fatty acids can really be used for oxidation. Next, a CoA molecule must bind to the carboxylic end of the free fatty acid, turning the molecule into an acyl-CoA. Don't be confused with acetyl-CoA. Acyl-CoA is a fatty acid that has an OH, that has its OH hydrophilic head replaced or attached to CoA molecule, and you can see that here actually, right? So this is a fatty acid chain. This would be normally the head, and now this OH is just replaced with the CoA right here. Okay, so acyl-CoA is different than acetyl-CoA. Lastly, the acyl-CoA must be transported into the mitochondria of the cell to begin oxidation. Just like with the Krebs cycle, beta oxidation occurs in the mitochondria. So it must be transported there. The first step is to prepare to convert the acyl-CoA into an enoyl-CoA, which has a double bond between the alpha and beta carbons. This step is an oxidation step, and like I mentioned last time, whenever you have an oxidation step, that means something else must be getting reduced. Well, the thing we usually want to get reduced is called either FAD or NAD into FADH or NADH. In this case, it's FAD into FADH. And this is what an enoyl-CoA looks like. So if you remember, the acyl-CoA just looks like a fatty acid chain, whereas now we have this double bond in between this alpha carbon The alpha carbon is the one is the first carbon next to this acyl carbon, right? So this carbon doesn't actually count. This is the alpha carbon. And then this is the beta carbon. And the reason it's called beta oxidation, and you'll see, is because the beta carbon is very important specifically. But first thing we do is we form a double bond between this alpha and beta carbon. And this produces FADH2, which is very useful. The next step is a hydration step, which adds an OH group to the beta carbon. So we break this double bond, and instead we add into this molecule water, H2O. That's why it's called a hydration reaction, because we're adding in water as opposed to a dehydration reaction, which we remove water. We add in water. The OH from water goes onto this carbon, the beta carbon, and then the H from the water, the remaining H, goes onto this alpha carbon. So now they're both, again, fully saturated, but now instead of having an H right here, it now has an OH on this beta carbon. The reason for that is because, like I mentioned, we want to make acetyl-CoA from this really long chain. What we can do, since acetyl-CoA is a 2-carbon molecule, we have to cut it like this at this carbon, and we have to cut. And this will be acetyl-CoA, and this will be the rest of the fatty acid, as shown by this R group. In order to do that, in order to make this a fatty acid, you again have to make an O here, right? A double bond O. Otherwise, it wouldn't be a fatty acid. It would just be a really long hydrocarbon chain. So our goal is to convert this beta carbon into an acyl carbon, right? And the first thing we have to do is we have to make a double bond and then add in water to make it an OH. From here, the products of beta oxidation are 1 NADH, 1 FADH. Oh, wait, I believe I skipped. Yeah. So the next steps, the third step is oxidation of the beta carbon into a ketone instead of an alcohol. So again, I mentioned we have to make an acyl carbon, which is a double bond O. So right now we have an alcohol, so we need to make that into a double bond O. This is called the ketone, right? And again, we're oxidating alcohol into ketone. This uses NAD as a cofactor, turning it into NADH. The last step is then simply cutting off the alpha carbon and the acetyl carbon as acetyl-CoA, right? Again, 2 carbons, the acetyl carbon and the alpha carbon get cut off. Together, they already have the CoA attached to it on the end, so already made as acetyl-CoA. This shortens the free fatty acid chain by 2 carbons, releases acetyl-CoA, and allows the cycle to begin anew. Again, we can run through this cycle again because we now have another fatty acid. We started off with, say, a 16-carbon fatty acid. Now we have a 14-carbon fatty acid plus acetyl-CoA. And we can now start again with the 14-carbon fatty acid, go through another cycle. Repeat all of the steps again. Now we have a 12-carbon, back 10, 8, 6, 4, and eventually we'll be left with only 2-carbon acetyl-CoAs, and that's it. So the products of beta oxidation are 1 NADH, 1 FADH2, and 1 acetyl-CoA per cycle. The number of cycles depends on the number of carbons in the free fatty acid chain. Most consumed free fatty acids that we eat have either about 12, 14, or 16 carbons in the chain. So there will be 5, 6, or 7 cycles needed to fully break them down, respectively. And if you're ever given a number, the way to do this easily is, say you had a 12-carbon chain, you have to go into 10, then 8, because we're going down by increments of 2, then 6, 4, then 2, and then we can't go down to 0. We're done at 2. This has 1, 2, 3, 4, 5 cycles, right? Each of these arrows represents going through the entire cycle once. So we go through 5 cycles. If I added 14, if we start off with 14 length chain, we would just have to add one more cycle, 6, 16 would be 7. Okay. And it's not necessarily as crucial to know as it is for the Krebs cycle, to know the exact number of ATPs that you produce per beta oxidation, because beta oxidation also does require actually taking in a little bit of ATP to make all the necessary preparations to begin it. But it's not exactly crucial to know the exact number of ATP total that you're making. Just know that it's a lot more than it, for example, it is for glycolysis or glucose, because you are making multiple acetyl-CoAs. The bare minimum you can make is 5, 6 acetyl-CoAs compared to the 2 you would make in glycolysis if you were breaking down a glucose molecule. So it's much more efficient. And most triglycerides, like I mentioned, will have 3 fatty acids. So that could be 15, 18 just from one molecule.